After learning about prokaryotic cells in the previous lecture, the third lecture, we will now talk about the eukaryotic cells. Now, eukaryotic cells, the main prime difference from prokaryotes is that it has a proper, well-organized nucleus along with cell organelles. Now, let us see the other details. Now, eukaryotic cells. So eukaryotic cells, these are the different types of eukaryotic cells that we see around us. They can be seen in plants. All plants are made up of eukaryotic cells. All animals are made up of eukaryotic cells. Then the fungi, all fungi, they are made up of eukaryotic cells, as well as the protists, the protista members. Now protista members are unicellular eukaryotic structures. These are protista. Earlier they were classified under animals, but later, seeing the differences from the proper animals, they have been given a different status, different kingdom totally, that is kingdom protista. Likewise, the fungi, earlier they were placed under plants, but as they are purely heterotrophic, they are not autotrophic, so in no way they can be compared with plants. So that is why even fungi, they have been placed under a different kingdom, that is kingdom fungi. So in general, that 5 kingdom classification says where one of the characteristics is also the type of cellular nature, that is cells, 5 kingdoms are there. One is kingdom Monera, which includes only prokaryotic cellular structures. The rest are kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. So the rest 4 kingdoms all have eukaryotic cellular structure. So now we will try to learn the basic things about the eukaryotic cellular structure. Before going into the details of it, let us try to know some differences between the plant and animal cells. Now, plant and animal cells, many of the features are common. The common features are presence of mitochondria, that is the energy yielding cell organelle, Golgi apparatus, the cell organelle which helps in packaging of proteins and lipids, rough and endoplasmic reticulum, which helps in formation and transport of substances, then nucleus, which contains the genetic material, the hereditary material, then the cytoplasm, the gel-like fluid which is present, which fills the cell, and ribosomes, the protein factories of the cell. So these are the organelles which are found common in both plants and animal cells. They are common in all types of eukaryotic cellular structure. Now, some features will be present only in plants, absent in animals. Some will be present only in animals, but absent in plants. Like say the cell wall, which only plants have. Also fungi, they have the cell wall. Fungi and plants, they have cell wall around their cells. Animals, they have no cell wall. They are purely devoid of cell walls. Then plant cells, they have a very large vacuole, whereas animals, they have small or almost very insignificant vacuoles. Chloroplasts, which contain chlorophyll, are exclusively present only in plants. The basic character which defines plant is the presence of chloroplast. So that is why they are able to prepare their own food, that is, they can photosynthesize. Animals, they are devoid of chloroplast. Then flagella, if present, it will be only in some gametes. Animal cells may have flagella, like say when we talk about some unicellular forms, they will have flagella. In animal cells also, some structures, some specific organ cells will have flagella, like say the male gametes, they have a long hair-like structure which will help in movement. Other than that, one more cell organelle, that is centrosome, which is present in animals but absent in plants. This is the figure of plant cell and this is the figure of animal cell. So plant cell, you can see as the cell wall is present, so it has a specific shape, whereas animals, they do not have a cell wall, concrete cell wall. As such, the shape can vary. Then other than that, the plastids or chloroplast present in plants, absent in animals. They have a large vacuole. They have a very insignificant or tiny vacuole. So, and here they have a centrosome which is absent in plant cells. Other than that, the other organelles remain common. Endoplasmic reticulum, Golgi bodies, mitochondria. About all these organelles we will learn in our upcoming lectures. So, the other features, everything remains the same or remains common in both plants and animals. Even with regard to function also, it is the same. Now we will go into the cell membrane. The cell membrane is the outermost covering of cells. Okay, it is the covering which holds the cytoplasm, that is known as the cell membrane. Covering. That covering is semi-permeable, means it allows only partial movement of substances. It will allow the movement of some constituents which are required and some which are not required. Okay, that means it will help in selective movement of minerals or chemicals into and out of the cell, hence known as semi-permeable. Now, a fluid mosaic model was proposed by Singer and Nicholson in 1972 about the cell membrane. They said that the cell membrane is made up of two layers of lipids, two layers of fats. Here is the one layer and here is the second layer. Interspersed or in between is the presence of protein. In between is the presence of proteins which may be throughout the cell membrane or may be present just on the surface. Accordingly, we have integral protein and peripheral protein. Other than that, they may have some carbohydrate chain as also some fat chains. That was the structure proposed by Singer and Nicholson on the cell membrane. They said that the lipids, these lipids which makes up the two surfaces of the membrane, they are bipolar. They are bipolar means they have two ends. One end which will, this is the layer is bipolar. So each lipid molecule has two ends. One is polar and the other is non-polar. The polar end will always be oriented towards wherever there is presence of water, and the non-polar will be away from water. Now, the external surface of the cell definitely has some tissue fluid, extracellular material. So, extracellular material which contains water. So, what will happen? These polar molecules, these polar lipids, the polar end will be oriented towards the water containing solution. Whereas, similarly, the other lipid molecule will also orient itself like according to the presence of water. Inside it's filled with cytoplasm, so it will face the polar end will face towards the cytoplasm which contains water. Let us see few more points. It is and hence it is called as quasi-fluid nature, that is semi-fluid nature. Lipid bilayer with protein. The proteins can be two types: integral protein, peripheral protein. Integral, when it is throughout it is present, and wherever protein is present, there you will find absence of the lipid layer. That means there is break in the lipid layers. Then here furthermore we have the same structure. This is the bilipid layer. Okay, the yellow points, the yellow heads are the polar end, that is the hydrophilic end. They are facing, this is the external surface outside of the cell membrane, this is inside of the cell membrane, cytoplasm. Here also water is present, even inside water content will be there. So what will happen when you place one polar molecule, you just place it here, you place in any direction, what will happen? It will automatically orient itself towards the presence of water, the hydrophilic end. So hydrophilic end faces here, whatever lipids come here, they will face inwards. The hydrophilic end will face inwards. So consequently, the hydrophobic ends, tails, will be one upon other. Okay, they will remain in this manner. Why? why is this arrangement? This arrangement is to prevent any and every substance from passing through the cell membrane, because lipids, as you know, they are water repelling. They will repel other substances, water containing substances, to pass through the membrane. They will restrict the entry of substances. In between, as I already told, there are the proteins. These proteins are called as transport proteins. Why? Because it is through these proteins that minerals or substances will pass. That means they act like tunnels. Okay, they will act like tunnels or channels. Two types: integral ones, okay, integral ones, or the globular ones, which will be present just on the surface, the peripheral proteins. Besides that, carbohydrate chain, lipid chains, they are attached, which will give additional support to the cell membrane. So a cell is completely surrounded by such a surface. So here again, in water, interactions of the hydrophobic tail and hydrophilic heads generate a phospholipid bilayer, which is 2 molecules thick. 2 molecules thick. What are the molecules? Lipid molecules. Head group or hydrophilic end will face outward and inward. Okay, for the other layer it will face inward. Tails are packed together in the interior. Then functions of the cell membrane, they help mainly in transport of materials. There are two types of transport walking, that is active and passive. Active will be which requires energy, and passive will be which does not require energy. So passive transport is comparatively very simple. It is simply a physical movement, the physical movement of substances as per the concentration gradient. Higher concentration to lower concentration. Suppose outside the cell there is high concentration of sodium or phosphorus or any mineral. Inside the cell, minimal quantity. Then what will happen? Spontaneously, just physical movement through the protein channels will take place. Simply, it is a physical process. But sometimes the cell needs to move elements or substances against the gradient. That means it has to move from low concentration to high concentration. So in such cases, there will be utilization of energy by the proteins, the channel proteins, the transport proteins. Then such a movement will be called as active movement or active transport. So for passive transport, the main mechanism is simple: diffusion from high to low concentration, which can also include osmosis, that is water movement. But for active transport, there is always involvement of energy, utilization of energy in the form of ATP. Next, we move on to cell wall. Now, cell wall is also a surface that is found covering the cell. In what type of cells? Yes, it is in plant cells, as also in fungi. Even in fungi, we find cell wall. Also, if you can recall, cell walls are found in prokaryotes, in bacteria even. So what is a cell wall? It is a rigid structure that gives support to the cell main function. Okay, they are made up of different substances, but in plants they are mainly made up of cellulose. Cellulose is a polysaccharide. Polysaccharide means a carbohydrate. Many glucose molecules will unite together to form a polysaccharide, cellulose. That makes up the cell wall. Animals do not have cell wall. So here it is that onion peel cells. So here this blue. Okay, so here you can see these are the cell walls. Okay, this is a microscopic view given. Furthermore, even here, this is an under a high-powered microscope, we can see the cell. Okay, this is the cell, the internal structures, plant cells. These are the chloroplast, which is making the entire cell green, and outermost is brown layer, that is the cell wall. They have cell membrane inside, and outside they have the cell wall. Cell wall materials can vary. Okay, like say in fungi, it is made up of chitin, which is also a type of carbohydrate. In bacteria, as we already saw, it is made of peptidoglycan, and in plants, it is made up of cellulose. So what are the three substances making up cell wall? Fungi is made of chitin, peptidoglycan makes up bacterial cell wall, and cellulose makes up plant cell walls. Now the cell wall, okay, furthermore, we can see here, these are the individual cells. These are the cells, and here the broad part has been shown as the cell wall. Now two adjacent cells are always, they have a common dividing layer. That common dividing layer is called as the middle lamella. Okay, these two adjacent cells, or these two, they have a common layer in between called as middle lamella. Inner to the middle lamella is present the secondary cell wall, okay, and outer to the secondary cell wall we have the primary wall. So middle lamella, primary wall, and then the secondary wall. This blue layer that is given here, that represents the cell membrane. So here further it can be shown, okay, a diagrammatic view you get here. So here you have the middle lamella, okay, you have the primary cell wall, here the primary cell wall, plasma membrane, this is the plasma membrane. Plasma membrane and cell membrane are the same things, okay, they are the same. Two terms given, cytoplasm, nucleus, all these are the inside, the interior of the cell. So here you have the secondary cell wall, you have the plasma membrane, okay, the first is plasma membrane, then the secondary cell wall, then you have the primary cell wall with the middle lamella. Here is the middle, this thick line is the middle lamella. So this is the middle lamella, this primary wall, okay, primary wall, and then here you have the secondary wall. Okay, so middle lamella, primary wall, secondary wall, plasma membrane for the cell this side, and for the cell the other side, middle lamella, primary wall, secondary wall, plasma membrane. So that makes up the cell wall. Sometimes one more layer is deposited. Sometimes, okay, especially in cells which have undergone like aged, or they have aged, or like say they have matured very highly and they need some additional support, then a tertiary cell wall is also present. It is considered to be dry residue of protoplast. So where will it be present? It will be present internal to the secondary layer. Then plasmodesmata. Now, two cells can remain interconnected in spite of the presence of cell wall. Some tiny pores present on the cell wall are called as plasmodesmata. Through the plasmodesmata, the cytoplasm of two adjacent cells can remain interconnected. As you can see here, they are interconnected. Why? To facilitate the movement of substances from and to the cell, or from and to the cell. Then plasmodesmata further it is shown here. Okay, so when you we have a look. So here there's a cell wall. You can see the cell wall is ruptured here. Okay, the cell wall, it is showing breaks. See here, the cell wall, okay, it is not continuous. Here some breaks are present. Some breaks are present. These breaks are called as plasmodesmata. Even in the other figure, it's given here, enlarged view, which will allow movement. Of course, these pores are very, very tiny. They are only 40 to 50 nanometers in size. Now finally coming to the functions of cell wall. The main functions are to provide strength to the plant cells. Okay, then it helps to protect itself against some external solutions. External solutions which may be dangerous for the cell. It will help in protecting against such mediums. Then it gives shape to the cell, and because of the cell walls, they can withstand the changes that are happening around, the environmental changes. Then also it protects against injuries and pathogens. So in a nutshell, what are the functions of cell wall? To provide strength, to give shape, to protect the cell against pathogens, mechanical injury, as well as chemicals. So these are the main functions of the cell wall. So in this lecture, we learned about the differences in plants and animal cells. we learned about the plasma membrane or cell membrane, the structure of it, which remains common in both plants and animals cells, and we learned about cell wall, the details of cell wall in plant cells. So in the upcoming lectures, we will move into, or we will discuss about the different types of cell organelles that exist in cells. Thank you.